In this slideshow, we will consider Mendel's hypothesis explaining the 3-to-1 ratio inheritance pattern observed in the F2 offspring. It was Mendel's quantitative analysis of the F2 plants from thousands of genetic crosses that allowed him to deduce two fundamental principles of heredity, now called the law of segregation and the law of independent assortment. In the development of his hypothesis, Mendel considered four related concepts, and it is these concepts that enable us to know now what we do about genes and chromosomes. First, alternative versions of genes account for variation in inherited traits. For example, the gene for flower color in pea plants exists in two versions, one for purple flowers and the other for white flowers. These alternative forms are called alleles. Note that a capital P represents purple allele, while a lowercase p represents the white allele. The illustration here shows a pair of homologous chromosomes in an F1 hybrid pea plant. Each gene is a sequence of nucleotides at a specific locus along a particular chromosome. The paternally inherited chromosome, shown here in blue, has an allele for purple flowers, which codes for a protein, an enzyme to be exact, that controls the synthesis of purple pigment. The maternally inherited chromosome, shown in red, has an allele for white flowers, which results in no functional enzyme being made. Second, for each trait, an organism inherits two alleles of a gene, one from each parent. Remember, each somatic cell in a diploid organism has two sets of chromosomes, one set inherited from each parent. If the two alleles at a particular locus are identical, they are referred to as homozygous. Or if the two alleles differ, they are referred to as heterozygous. So to use the letter P, as we did on the previous slide, a plant that has two purple alleles is homozygous for purple and is represented with two capital P's. A plant that has two white alleles is homozygous for white and is represented with two lowercase p's. Or if a plant has one purple allele and one white allele, then it is heterozygous for purple and is represented by a capital P and a lowercase p. Thirdly, if the two alleles at a locus differ, then the dominant allele determines the organism's appearance, while the recessive allele has no noticeable effect on the organism's appearance. And as mentioned earlier, Mendel concluded that the F1 hybrids had purple flowers because the allele for that trait is dominant and the allele for white flowers is recessive. The fourth and final part of Mendel's model, the law of segregation, states that the two alleles for a trait segregate, or you can think separate, during gamete formation and end up in different gametes. Therefore, an egg or sperm cell only gets one of the two alleles. And upon fertilization, the resulting zygote then receives a pair of alleles, one from each parent. To give an example of how this works, consider a heterozygous purple flowered plant. Remember, to be heterozygous, it means the hybrid plant has one dominant allele and one recessive allele. Following the law of segregation, this means that the dominant allele separates from the recessive allele during meiosis, resulting in 50% of the gametes containing the dominant allele and the remaining 50% of the gametes containing the recessive allele. A helpful tool to illustrate all of the possible combinations of sperm and egg in the resulting zygote is the Punnett square, which was a tool devised by Mendel. Let's review the law of segregation on the next slide, as well as demonstrate the significance of the use of the Punnett square. This illustration shows the cross we reviewed in the first slideshow of a purple flowered plant with a white flowered plant. But let's review the experimental setup and results once again using the concepts and vocabulary we have learned since. Recall, the parent generation is true breeding. Now that we have talked alleles, and remember, we are using upper and lower case letters to represent dominant and recessive alleles respectively, the purple flowered plant is homozygous dominant for purple, meaning that it has two dominant alleles, or two capital P's. The white flowered plant is homozygous recessive for white, meaning that it has two recessive alleles, or two lowercase p's. Now, when each plant produces its gametes, notice that the purple flowered plant can only pass on the dominant allele, while the white flowered plant can only pass on the recessive allele. Therefore, all offspring in the F1 generation will be heterozygous for purple flowers. Now, when the F1 plants produce gametes, note that half of their gametes will have the dominant allele, while the other half will contain the recessive allele. And remember, when Mendel crossed two of the F1s together, he got a ratio of 3 purple flowered plants for every 1 white flowered plant. But let's use the Punnett square to explain these results. Each square inside represents the possible combination of alleles in the offspring, with the contents of the square equaling 100%. To set up the square, we list all the possible gametes from one parent on the left side of the square and all the possible gametes from the other parent along the top. Then just bring down the alleles or the letters from the top, pairing them with the alleles from the left to yield all possible combinations in the offspring. In the case of the heterozygous allele combination, it is best practice to always write the dominant allele first in the pair. Given that purple is dominant to white, note the 3-to-1 ratio occurs because the upper left square is homozygous dominant for purple, the upper right and lower left squares are both heterozygous for purple, while the lower right square is homozygous recessive for white flowers. Thus, this accounts for why Mendel obtained the 3-to-1 ratio in the F2 generation. Now keep in mind that this is the same thing as saying that there is a 75% chance of getting a purple flowered plant or a 25% chance of getting a white flowered plant when crossing two F1 plants. It does not mean that if 4 plants are produced, that automatically 3 will be purple and 1 will be white. As you may have noticed in the Punnett square on the previous slide, there were 2 different allele combinations that both resulted in purple flowered plants. Both the homozygous dominant and heterozygous combinations result in purple flowers. So because of the different effects of dominant and recessive alleles, an organism's traits do not always reveal its genetic composition. Therefore, we distinguish between an organism's physical appearance, called its phenotype, and its genetic makeup, its genotype. As shown here, the 3-to-1 ratio that we mentioned previously is actually the phenotypic ratio. If we consider the allele combinations possible in the offspring, notice that that is a bit different. The genotypic ratio, or the allele combinations, for the same results is 1 to 2 to 1, meaning then that there is a 25% chance of having plants that are homozygous dominant, a 50% chance of having heterozygous flowers, and a 25% chance of having homozygous recessive flowers. So as we proceed with working with Punnett squares, keep in mind that you will have to determine both phenotypic and genotypic ratios for the squares you'll be working. So here's a question for you now. If I show you a purple flowered plant, you obviously know that the phenotype is purple. But what is this plant's genotype? And remember, there are two options. Either the genotype is homozygous dominant or heterozygous. But you cannot tell just by looking at the plant. So how do you determine the genotype of the purple flowered plant? Well, Mendel devised yet another tool called the test cross in order to determine the genotype of an organism showing the dominant phenotype. In a test cross, the individual with the unknown genotype is crossed with a homozygous recessive individual expressing the recessive trait. So for this particular example, the purple flowered plant is crossed with a white flowered plant. The Punnett squares shown here show the two possible outcomes of the cross. If all of the offspring have purple flowers, then the purple flowered plant must be homozygous for the dominant allele, because a homozygous dominant crossed with a homozygous recessive produces all heterozygous purple offspring. But if the purple and the white phenotypes appear among the offspring, then the purple flowered plant must be heterozygous. The offspring of a heterozygote crossed with a homozygous recessive are expected to be 50% purple and 50% white. And in fact, even if only one white flowered plant results, then the mystery purple plant must be heterozygous. It was this same approach that enabled Mendel to determine the unknown genotype of his plants expressing the dominant trait.